I'm pleased to introduce Dave Carlson. Dave is one of the newest hires here to DCRI, though he is no stranger to Duke. He's done his undergrad, master's, PhD here. For some reason, he left to go to Columbia to do a postdoc. It left such a bad taste in his mouth, he came back to do another postdoc here, all in electrical engineering, computer science. He's since worked a lot with psychiatry. He does a lot of work around deep learning, neuroimaging, natural image processing you're getting into now. Yep. And not only, so he is a joint hire between biostatistics and bioinformatics along with chemical engineering. He also has a secondary, civil environmental engineering. He has a secondary in the statistics department, and we are very fortunate to have some of his attention here in DCRI as well. Thank you.
Thanks.
Maybe don't do that, sir.
I don't think so. Thanks so much for the introduction. So today I'm going to be talking about interpretable machine learning to deconstruct the neural basis of psychiatric disorders, which I hope everyone finds to be a lot of fun. I think it's fun, so hopefully everyone agrees. So I just wanted to expand on my background just a little bit. So as Ben mentioned, I'm a very recent hire. I started July 1, so this is, I guess, just the end of my 4th month here. And I was hired jointly between the Pratt School of Engineering and also Biostatistics and Bioinformatics, of which that appointment is here in DCRI. And so one of the things that I really care about is how can we use machine learning and how can we make it useful to understand scientific and health data? So a lot of what's going on in machine learning is we use it to make predictions. So there's been a lot of use of machine learning in creating things like risk scores, determining who's at risk. But one of the things that I fundamentally care about is how can we not just do accurate predictions, but also try to understand how it's making these decisions and why it's making these decisions. And so as Ben mentioned, I have my PhD from Duke in electrical and computer engineering. I had some postdoctoral training in statistics and data science at Columbia and also at Duke in electrical and computer engineering and also psychiatry and behavioral sciences. So I think at this point, everyone has some idea of what's going on in machine learning. I don't think you can open up any big newspaper recently and not see an article on some sort of machine learning application. But just to make sure that we're all on the same page, I wanted to give an operant definition of what's going on. And so one of the definitions that's commonly used is that machine learning is a statistical subfield of artificial intelligence that gives computers the ability to learn without being explicitly programmed. So the idea of machine learning is that we're not trying to make right how these decisions should happen. We're just going to give it a lot of examples. We're going to give it examples and say, here was a case. Here's a lot of background data, for example, health information or some other sort of measurements, and here is the outcome. And what we want to do is say, well, now we have some structure, and we're going to learn how to make these decisions just by feeding data to these meta-algorithms. So we're going to actually learn the algorithms that we're using to make decisions from data. And so I think everyone in this room is benefiting from machine learning approaches daily, whether you realize it or not. Email is doing a lot better with spam filters. These are all done on machine learning. All the networking happens with machine learning algorithms. Image enhancement, so including a lot of the medical devices actually in use at the hospital right now, a lot of the image enhancement is actually done using machine learning every time you take a picture on your smartphone. So it's really kind of pervasive today, whether you realize it or not. And so as I mentioned, one of the things that I really care about is how can we actually take machine learning and make it very useful to actually understand what's going on, not just make predictions, not just clean data, but actually use it to understand the scientific underpinnings of what's going on. And so we're going to talk about how we can do this by trying to model and using machine learning on trying to understand the neural dynamics of psychiatric disorders. And so most of my talk is actually going to be on animal models. And one of the really nice things about animal models is whenever we're learning things with our algorithms, we're actually going to be able to run causal validation. And at the end of the talk, I'm going to talk a little bit about the efforts we're doing on human studies. And so we're doing some secondary analysis of some clinical trials that are being run through the Autism Center. And so I wanted to make sure that it's on Halloween. And so now that I've given my background, we have 2 pumpkins here, and I will tell you one of these I carved, and one of these my wife carved. And so if you have any guesses, you can tell me after the talk. But wanted to make sure I got that in there. And before I actually dive into going into how we're using this in our particular application in psychiatric disorders, I'm really curious about how pervasive machine learning has been within people's work at DCRI and at Duke. So one of the first polling questions I have is, has machine learning visibly impacted your work at Duke? I talked first about how there's been a lot of background pervasiveness of machine learning, but I'm really curious how visibly impactful it is on people's work here. So far it looks like everyone has some impact from it, which is about what I was actually expecting more people to say no, but I'm kind of glad to hear that everyone at least realizes it's being impactful to what's going on in their own work, although apparently only with minor impact. Cool, that's actually about what I was expecting. So, although I'm making inferences too quickly. Got to wait for all the data to come in. You're underpowered, you know. All right. Well, that's actually very interesting. That's about what when I first created this question, I actually thought it was going to be more on the no section. But then after talking to a few people, they were like, no, everyone's starting to talk about this now. And so now that we've done that, I wanted to dive into this particular application that I was talking about, and how we can actually try to use machine learning to do data-driven science. And so as I mentioned, we want to use this data to create scientific understanding. We don't want to just run these algorithms on this data to create predictions. We actually want to generate scientific understanding so that we can understand the problem. And so a lot of the work that I'm going to talk about at first here is joint work with the Laboratory for Psychiatric Neuroengineering, and it's led by Kafui Daraza, who's shown there on the right. And one of the questions that we've been trying to analyze together and answer together is, how can we analyze, or how can we turn this vast amount of brain information data into actual interventions for psychiatric disorders? For people that aren't familiar with what's going on in neuroscience, there's been a lot of efforts from the BRAIN Initiative led by NIH that has really created transformative technologies such that we're now recording much larger scales of information than was ever previously possible. So we really want to say, well, let's not just get massive amounts of information. Let's turn this massive amount of information into actual treatment on psychiatric disorders. And so the Laboratory for Psychiatric Engineering has some interesting capabilities. And so one of the things they're doing is they're actually collecting neural data from multi-site electrodes in rodent models. So almost everything in the beginning here is going to be talking about a mouse model of depression. And when we say multi-site electrode, what this means is these are electrodes implanted inside the brain, but they're implanted in many different brain regions. So you have many different brain regions that are implanted. And so we can actually get a lot of information from many different areas of the brain, closer to a whole brain dynamics that we're interested in. And we're actually going to, if we can come up with theories of how this information is changing, how these patterns are changing in ways that we think are related to the outcomes, one of the really nice things is that they actually have very nice facilities for doing something called optogenetics. I'll explain a little bit more of optogenetics later, but what optogenetics is it's a neurostimulation technique. So we can actually stimulate the neurons in order to modulate the brain activity. So if we can figure out patterns that we think are going on, we're actually going to be able to run causal validation of the learned trends that we're finding. And the end goal is to try to create treatments for psychiatric disorders. So if we're going to talk about a mouse model of depression, it's very important to understand how you can actually tell whether a mouse is acting with depressive-like behavior. So one of the common ways that's done is to measure something called anhedonia. Anhedonia is basically the lack of pleasure. So things that should be providing you pleasure, you're no longer experiencing pleasure. And so some of the simple ways this is measured is things like sucrose preferences. And what this means is that mice typically prefer sugary snacks and sugar water. And if they're not preferring the sugar water, this is a sign that they're no longer enjoying it. So you can measure how much they prefer sugar water to like normal water. You can also look at reduction in sleep patterns. You can look at whether they're still enjoying exploration, or if you put them in a new environment, whether they explore at all. You can also look at sex drive, which I've fortunately never had to check myself. And, but there's also a lot of more direct tests. So the anhedonia measurements are usually taken over several days. So it's an accurate measurement, but it takes a while to do. But there's something called the tail suspension test. And the tail suspension test is not a new test. It's actually been used to validate antidepressants historically. So a lot of the antidepressants now used in market were originally shown to be effective in a rodent model via the tail suspension test. And it's a lot easier to see what's going on through an experiment, through just showing the video. And so I apologize if this is disquieting to anyone that's currently eating. So what we have is we have a mouse. The mouse is suspended by its tail. As you can see, the mouse is actively trying to escape. This is actually considered a good sign for this test. If the mouse becomes stable like this for long periods of time, it's considered defeated. This is a sign that it's given up. So inactivity is actually considered a very bad sign in this test. Now I wanted to point out, we have a wire that's coming down here. This wire goes and connects to a head stage. This head stage is connected to those electrodes implanted in the brain. So while this test is running, we're actually getting live readouts of neural activity that's going on in the brain. So how do we actually go through and get this information? Typically, you get this information through electrodes. So these types of electrodes are actually physically implanted inside the brain. You get a variety of these electrodes, and we place them in many different brain regions. They're pretty scary-looking devices, but each one of these little pins out here is a different electrode, and so you can get a large scale of information from each of these areas. And what we're getting is we're getting two different types of signals. The first type of signal that people often talk about is something called neural action potentials. Neural action potentials are, if you think of an individual neuron, it's when an individual neuron spikes. And so what this is typically represented by is individual neurons and their spike trains. And what a spike train is it's a binary time series which says at each point in time, did this neuron spike or did it not? So what we have here is we have a lot of horizontal trends. Each one of these different lines is an individual neuron. And you have a dark mark when that individual neuron fired or did not fire through time. And we color-coded them by region. So you can get a lot of different individual neurons out of these signals. And I'm going to talk about neural action potentials a little bit later. But I want to get into something called a local field potential. A local field potential is low-frequency oscillations of your neural activity. If you look at studies that are done through EEG, which is a much more common experimental type in humans, you're basically measuring local field potentials, but you're measuring them at a much lower fidelity than implanted electrodes. You can think of this as a much, much higher fidelity EEG type of measurement. And what it is that for all of the electrodes within a given region, we take all those electrodes and we average the signal, and you get out these time series that are your low-frequency voltage oscillations. And you get one per brain region, and they're pretty small, but we can zoom in on them. Oh, I wanted to mention two other things. The neural action potentials are actually really fantastic because they're the smallest fundamental unit of information processing in the brain. However, mapping neurons from individual to individual is incredibly difficult. So if you want to study a population level effect, so if you want to study how things are changing on the scope of a population as it goes through a neurological disorder, you're a lot better off using these lower fidelity measurements, which a local field potential is, that's actually going to be much more stable across the population. So if we look at what's going on in these local field potentials, we have 4 different brain regions there. We have the nucleus accumbens, the ventral tegmental area, prefrontal cortex, and the amygdala. And if we zoom in on these 2 different signals here and we take just a half-second sequence of them, what you see is you see a lot of these low-frequency oscillations. Now, one of the things that you can probably observe is that these signals are highly correlated. And what I mean by that is that they have a lot of the same patterns. You see this low-frequency wave, and it goes up and down between these 2 brain regions at about the same rate. So one of the classic ways of analyzing this signal is to look at something called spectral features. So these spectral features are going to be frequency bands. So what we're going to do is we're going to limit this to, say, looking at just the 5 hertz frequency band or the 6 hertz frequency band. And within that frequency band, we want to know, are these signals power and phase coherent? And what that means is when the power in that frequency band on one brain region goes high, does the other one go high as well? And it turns out power coherence is a signal that these brain regions are communicating on that frequency band. And we're also going to talk about phase coherence or a temporal offset. So this is the idea that if you limit yourself to a frequency band, that when you have a temporal offset in that frequency band, you can find the best temporal offset that maximizes how similar the signals are. And this temporal offset that maximizes the similarity is going to help provide us with information of which way we think information is flowing. So on the right here, we're showing this temporal offset. This is the best temporal offset between the prefrontal cortex and the amygdala. And what we're showing here is we're showing this as a function of frequency. We're showing the best temporal offset. So we've marked this 5 hertz band here in red. And the prefrontal cortex is leading the amygdala in the 5 hertz band, which means that in this red color, we think in the 5 hertz band that the prefrontal cortex is sending information to the amygdala. Now, we actually find the opposite trend in about this 10 hertz band here. So in the blue, you can see that we have information going from the amygdala to the prefrontal cortex. So what this means is that we've actually found a nice feedback loop where we have information going from the prefrontal cortex to the amygdala and then back in 2 different frequency bands. Now, I want to be very careful when I'm talking about this causality and this directionality. So what's actually happening here is that all we're simply doing is finding correlations. These are just statistical correlations. As everyone should know, correlation does not imply causation. However, correlations can often be very useful for figuring out what we want to look into. And in fact, in this particular application, there are numerous studies that show that this type of correlation often does imply causation. So while we can't say for certain from this observational study that this is in fact how information is flowing, we can use this as a very nice way of generating hypotheses. So we can use this as a hypothesis generation. Now, this is a classical way of doing this type of experiment. And so you can go through, you can collect all your data, you can create these simple metrics. But one of the things that's really kind of problematic is that if we look at this, most of the information, most of what we're observing here is actually not related to our behavioral outcome. The brain does lots of things. Right now I'm talking. If I was talking, it's going to be completely unrelated to my emotional state, right? Your brain does lots of things all the time. So we want to find the part of the information that is actually related to the behavioral outcome that we want, because that's what we're going to want to test on. So I'm going to talk about how we can actually pull out the subsection of information that's related to the outcomes in a little bit and how we can use machine learning techniques to do this. But I wanted to talk about, first, what do these things actually look like and how can we use them if we were in fact able to get them? And so what we're showing here is we're showing a complete or a multi-region brain network that we've actually found. This was from a stress paradigm in mice. And so this is actually related to your stress outcome. And so what we're showing on the left is going to be related to that power coherence. The left is going to tell us which brain regions are communicating, and the right is going to tell us how we think information is flowing. So on the left, what we have is we have this wheel. Each wedge of this wheel corresponds to an individual brain region. So we have, down here, we have the prelimbic cortex, and this is the ventral tegmental area, and so on. And within each wheel, we have 10, 20, 30, 40, 50. This corresponds to hertz. This is what frequency band we're looking at. And what you can see is that we have this blue line that connects these two brain regions at the 8 hertz bands. And so what this would mean is that at 8 hertz, the prelimbic cortex and the ventral tegmental area are communicating with each other. And we have blue lines and we have pink lines here that are connecting these brain regions. The colors are absolutely meaningless except to help us keep distinct frequency bands clear. So the blue ones are always hitting at 8, and the pink are always hitting at 15 hertz. So we can tell you within these particular frequency bands which brain regions we think are communicating. Now, in addition to tell you which brain regions we think are communicating, we can actually go through and try to predict how we think the information is flowing. So in blue, we start in a particular part of the hippocampus, and we send out to the rest of the brain there. Whereas in this pink band, which is a different frequency band, we actually get a feedback loop to the hippocampus. So if we can get these type of patterns, I can say, this is something here. This represents just a small part of the total information that we're measuring. This is only a small part of it, but this is the part of the network that we think is actually related to your behavioral outcome. So I can go and talk to a collaborator, say, this is exactly what we think is going on, and if we know what we have an idea, we can come up with a hypothesis of how we think this is happening. So as I mentioned, by working with collaborators, they've actually come up with a number of ways of doing optogenetics. So they can do neural stimulation. And so if we find these trends that we think are very important to our behavioral outcomes, not only can we say these are highly predictive, we can also say, well, let's come up with a way of testing it. Let's actually modulate your neural signals, and let's see if we get a behavioral response. And so what we have here is we have a mouse in the tail suspension test. And this mouse has been actually changed slightly so that it can have optogenetic stimulation. For those of you that don't know how optogenetic stimulation works, typically it's a viral method. So you inject a particular brain region with a virus. This virus injects individual neurons, and it makes these individual neurons susceptible to light. So in this particular case, if you were to hit that neuron with a blue laser, you would cause that neuron to fire. If you hit the neuron with any other wavelength of light, it's not supposed to respond. So we have here a yellow laser. The yellow laser is simply used as a control. So this is just so that you're matching the amount of heat that you're pumping into the brain region. So we have this mouse in the tail suspension test. We can pull out data in real time. And as we were talking about, what's really important in these patterns is actually the communication between brain regions. So we need to map what's happening in these neighbor brain regions so we can match the patterns. And then we can go and push through to a waveform generator, and we can actually stimulate the brain to try to upregulate or downregulate these neural circuits that we're learning. So there's a very simple question here. Great, we talked about a pretty complicated procedure. We have a complicated evaluation scheme. The question is, does it work? And since I'm up here talking about it, the answer is yes. And so can we actually go through and do this? And so this is a mouse going through the tail suspension test. This is actually a lot of mice going through the tail suspension test. And the tail suspension test lasts for 10 minutes. And we have our yellow laser that is our control laser. And on the y-axis, we have the percent time immobile. So the control mice are becoming immobile quite quickly. Whereas when we're actually upregulating these patterns that we're finding to be very important to the activity of the mice and the behavioral responses of the mice, we get an instant change in the behavior. They become much more resilient to this task, and they give up a lot slower than when we don't do this stimulation. And it's significant at the p equals 0.1 level. This is 15 mice total, 8 on the blue and 7 on the control. So there's also a lot of controls that I'm clearly not showing here. There's a lot of extra experiments to actually indicate that this is actually a thing that we think is real. One of the immediate questions is, well, if they're more active, are we simply upregulating activity, or are we actually making them more resilient? And so if we do very simple tests where we just check for activity, we get no differences. So we're hopeful that this is actually related to the things that we're trying to actually modulate, that we're actually modulating the underlying neural states of the brain and making these mice more resilient to these stressful situations. So I talked about what we can do if we can actually learn these patterns that are very important. So the question is, how can we actually go through and actually learn these patterns, right? How can we actually go and find these interpretable relationships out of this large amount of raw data? We can push all the data in we want, but if we can't find these patterns, it's not helpful to us. And so I will say this is the section where I usually go into a lot of math. And for this audience, I have cut most of it out. But if anybody is very disappointed by this and would like to talk about math more later, I'm always happy to talk about math. It's a lot of fun. So I was going to go through the basic ideas. So the first thing we do is we take our long time series. We have many minutes of activity for each mouse. And we have about 40 mice that we're going to talk about here. And we have each of our different brain regions. We have our local field potentials. And the first thing we do is we just window them. So we split them into 5-second chunks of time. And what we're going to say is that all the dynamics that we care about, all the changes in the system are going to occur on the scale of 5 seconds. Now, I want to note that the brain is changing much faster than 5 seconds. If you've ever reacted to something, I think you know that, or at least I hope your reaction time is less than 5 seconds. One would hope. But because we're dealing with mostly things like emotional states, we want to get a longer term dynamic here. So this has actually been a very robust amount of time to look at. And so what we're going to do is say, well, we're going to look at these, and we're going to consider each one of these an independent object that is statistically independent. And so the idea is that what we're going to do is we're going to follow something called a factor analysis approach. And the idea in a factor analysis approach is that if we have a very high-dimensional object, and these, in fact, are very high-dimensional objects. So each of our windows there, it's a matrix that's C by T. C here is the number of brain regions that we're looking at, which is about 10. T is going to be determined by our sampling rate and our, the length of time that we're dealing with. And so it turns out in these applications is about 1,000. So each one of our independent LFP windows is about a 10,000 dimensional object. So by most standards, that's a pretty high dimensional object. You can come up with higher dimensional things, but this is a pretty high dimensional object. So the idea of what's going on in factor analysis is we want to take this very high dimensional object and we want to represent it by a set of shared features or factors. And the idea is that if we have these shared features or factors, instead of having a 10,000 dimensional object, we're going to be able to describe this window by a very small set of objects, here about 20. And so the way this works mathematically is that we have our YW, and it's modeled as just being the superposition of these features or factors. So we have a WK there. This is going to be described as a kth feature or factor. And this kth feature or factor is going to be shared across all windows. And all we're saying is that in each window, we have this SWK. This is for the Wth window, how strongly is this kth feature or factor expressed? Is this kth feature factor very high, or is it 0, or is it very negative? And so you can describe what's going on in these windows by this. And then you also have something, this last term here, this epsilon term. This is just going to control model mismatch. It's modeled as additive Gaussian terms. You might call this a noise term. You might call this a background or unexplained biological variability. There's a lot of different terms for what you would call this depending on what field you're coming from. And I want to mention that if people have heard of principal components analysis, which I think a lot of people in the audience have, this is actually a very basic form of factor analysis. So this is actually a widely used methodology. This is not a transformative type of thing that we're doing here. But this is going to work really well for our application. And I just want to reiterate this. So we have a really high dimensional object here. It's about a 10,000 dimensional object. We're going to learn about 20 features. And once again, when I say learn those features, we don't know these features beforehand. We're going to have to learn what they are. And we're going to represent it. And so instead of analyzing that really high dimensional signal, once we know our features, we can describe what's going on in each one of these windows by those scores that say how strongly each feature is expressed. K here is about 20. So then we'll be analyzing a 20-dimensional signal rather than a 10,000-dimensional signal. And then all your basic stats start to hold really well, and you don't run into terrible multiple comparison problems. So the problem is that if you run through some of the classical approaches of doing this, such as principal components analysis, it actually doesn't work very well for this application. It turns out it doesn't predict very well, which is really frustrating. But it also is not very interpretable. If you've ever run principal components analysis, you can get out your principal components, but it's hard to make sense of what they're actually capturing. And so a lot of what our work has been on is how can we actually design these features or design these factors such that we can understand what's going on. And we know that we care about some particular things. So we know we care about these spectral or frequency properties. We know we care about power and phase coherence, or this directionality that I was talking about. We want to know what brain region is sending information to what other brain region. And the big thing is, in the end, predicting whether a mouse has depression is, I suppose, useful. But really what this is useful for is that we can actually start to develop hypotheses about how we could actually run experiments to actually understand the basis of what's going on. This is really not a prediction problem. We're going to set everything up as a prediction problem, but this is actually a case where we're trying to design an experiment to actually generate scientific understanding. So we really want to be able to interpret and develop hypotheses. And so I'm not going to go too much into what this is doing, but I wanted to describe what's going on graphically. So the idea that what we're doing here is that instead of having these factors or features done in a classical way, we're going to say, well, these factors or features are going to have to be defined by the properties we know we care about. And these factors or features are actually going to be allowed to change slightly from window to window, which is actually a very unusual thing. But as long as they have the same underlying properties, we're going to be OK. And what these underlying properties are going to be is what I showed you before. So we talked about these power coherence and these phase coherence properties, the directionality. So what we're going to learn here is actually these spectral phase and power properties that we know we can interpret and use to design experiments. But the end result is very similar. So we say if we have our window wide W here, then for this particular feature here, we can see how strongly this particular feature is expressed by this same factor score. So we're still going to use these factor scores in exactly the same way we did before. So really the change is just how we describe our features or our factors. And we're going to describe these in a very interpretable manner. But once we have these, we can still map to this 20-dimensional set of features. So we have a low-dimensional set of features. And then we're going to be able to interpret the features. So if we find one of these features is very, very important for our outcomes, we're actually going to be able to use this to understand what we think that feature is doing and use this to design interventions. And so for those of you that perhaps know some of the math, I will give one slide on what it's actually doing. And so how can we design these factor structures? And in fact, a lot of my contribution isn't on this slide that I'm hiding. So we have multiple publications on building the mathematical approach. Technically, it's something called a multi-output Gaussian process, and it's done through a multiple kernel learning framework in order to make this happen. And these are the same properties that I was telling you about before. We care about doing power coherence and phase coherence. But instead of going into the math and what's going on, I just want to show you that this is actually very helpful when we're actually going through and doing this mouse model of depression. So this is data that was collected within the Laboratory for Psychiatric Neuroengineering. We have 44 mice, and we have LFPs that were recorded during 3 different behavioral tasks. We have the home cage. This is where the mouse lives. It's supposed to be a neutral, calm environment. We have a novel environment, so the mouse is physically moved to a new environment. It's an environment that's unfamiliar to it, so it should be slightly stressful, but not anything like fearful or anything like that. And then there's something called a forced interaction test. So in the forced interaction test, you have this mouse in a novel environment, and then a larger threatening mouse is introduced to the environment. And the mouse you're testing on and measuring from is protected, but it may not feel protected. So it is actually a pretty frightening experience for the mouse. And we have control mice, but all the non-control mice are going to go through a chronic stress paradigm. So this is a common paradigm for inducing depressive-like behavior in an animal model. And we're going to learn 20 features or these networks, and we're going to try to predict 3 things. One is behavioral condition. Can we tell which behavior they're in? We're going to try to predict whether they've gone through this chronic stress paradigm or not. And the third thing we're going to try to predict is something called stress resiliency. So both humans and mice, if you put them through a chronically stressful situation, some people handle it just fine, whereas others actually start to have very negative effects and start having depressive-like behavior. And the exact same thing happens in mice. Some of these mice go through this chronic stress paradigm, and they're perfectly fine, and others are definitely not. And so it will be one of the things we want to do is, can we actually figure out who is resilient a priori them undergoing the stress paradigm? And so I wanted to show you what one of these features look like. So these 2 plots are just describing those properties I've been telling you about. To me, this is the less important part. And so what I want to show you is what these network scores actually look like, what these feature scores actually look like. So this is on the left part of this plot, we're showing prior to the chronic stress. And then on the right part, we're showing post-chronic stress. We have the control mice in black, and we have the resilient mice in green. The resilient mice are the mice that went through the chronic stress paradigm and were considered to behave normally afterwards. And the susceptible mice are in red. And these are mice that went through the chronic stress paradigm and were very, very susceptible to the chronic stress paradigm. And what happens is if you look at this, and we're showing in the bold, we're showing the averages of all the mice in that group. You can see that in the home cage, you already start to see differences, but you can see very clear differences in just how they respond to novel environments and other stressful situations. And post-chronic stress, they are actually very significant for how they're changed. But one of the things that's been very fascinating about this is not only are they very useful for predicting how you're doing overall, you can actually tell with very, very high confidence whether a mouse is going to be resilient to the stress paradigm or not prior to them undergoing the chronic stress procedure. So we can actually tell just from their neural signatures prior to them going through the stress paradigm, is this mouse resilient or is this mouse susceptible to chronic stress? And if you want a p-value, it's 9.7 times 10 to the minus 4, which is very good considering that this is only 40 mice. So we're very happy with that. We can look at a second predictive factor that's getting a pretty similar trend. And you can see once again, we get clear separations. We can also predict what's going on here. This is also statistically significant, but at a lower level. But I just wanted to recap what's going on so far. So we can use machine learning. And one of those things about machine learning is we're going to be able to decompose this really big, complicated signal into networks. But one of the big things that I really want to stress here is that we've been focusing a lot on how can we interpret these machine learning methods. Because if we can interpret them, we can actually go to interventions. And so there are currently ongoing efforts to test some of the things we've learned here to try to see if we can actually make mice more resilient. And we already have confirmatory experiments that have been run in early maternal separation. For those of you that don't know, early maternal separation is designed to mimic an early traumatic event. Early traumatic events in humans are known to make people that have experienced them more susceptible to depression later in life. And it turns out that these networks that we showed you that are actually upregulated in mice that are susceptible to stress, if you do an early maternal separation, all of these networks are upregulated. So we can actually see just in their brain signature that just by doing this early maternal separation that they're actually going to be susceptible to stress. Antidepressants hit on these networks that we found, which is really kind of fun. And so the last question here is, so this is great, right? However, this is DCRI. This is Clinical Research Institute. We primarily care about humans here, not necessarily animal models. And so how can we actually move to having an impact in human health? And so I wanted to briefly tell you about what we're doing with the Autism Center. And so we've actually adopted these techniques. So we created a lot of new techniques that were very useful for analyzing these multi-brain region local field potentials in animal models. But we can actually move to track the neural dynamics in human EEG studies. And in particular, we've been working with Jerry Dawson, who leads the Duke Center for Autism and Brain Development. They've been doing an early stage clinical trial on using a stem cell treatment in children that have been diagnosed with autism. And so what we've been doing here is clearly not a primary outcome, so this is all post-analysis of data that they've already collected. But basically, there's 25 children, and they have all been tracked for a year. And so we have two questions that we wanted to actually be able to answer. One of these is, well, if we have these children that have received this treatment, can we actually tell what in their brain signature has actually changed post-treatment? I would like to say as a result of this treatment, but we don't actually have the controls yet. The controls are coming soon. So I can only tell you what we can track in the patients that have received the treatment. And then the second question is, there's a lot of variation in how children respond to these treatments. It would be very nice to be able to tell you a priori, this treatment will be very successful, or this treatment should be avoided for this child, because it's not been uniformly successful for everyone, although on average it has been very successful. And so I wanted to point out, so instead of trying to track the dynamics as a time series, what we've been doing is actually been using a surrogate task of classification. So we have 0 months, 6 months, and 12 months post-treatment. So 0 months post-treatment is actually prior to the treatment. So this is actually our baseline result. And if we're trying to predict this, using these interpretable features, we actually get state-of-the-art accuracy. So we get 64% accurate. The classical approach people used was 50%. And there's been a lot of people across the country that have been working on using deep neural networks, which is a machine learning technique, in order to do this prediction, which is 58% accurate. And so it turns out that in this particular application, we're 6% more accurate, which corresponds to an increase of 2 standard deviations. And if you care about getting improved results, 2 standard deviations is a lot in order to improve your statistical power. And we can, once again, we can visualize these features. I can talk to the domain experts and say, well, this is the feature we found. Is this something that's useful? And this is a very useful thing because it turns out, especially in EEG studies, I don't know if anyone's familiar with them, but you can actually get a lot of spurious activity movement. So you don't want to find features that are just related to activity. So being able to say exactly what this feature is doing is very helpful. And there's something called the clinical improvement score, which is basically after the treatment, doctors evaluate each child and they assign a clinical improvement score based on the summarization of several different clinical scores. And this is just a measure of how much the child has improved. And it turns out that we can explain 30% of the variance of the clinical improvement score using just the information at the baseline. So we can actually get some information about why these patients are actually responding well, which these are things we're investigating into, whether they have known correlates in the literature. So where are we taking this? We're looking into a lot of additional confirmatory evidence. We're looking at doing more interventions in optogenetics and neurostimulations. And we have studies on working memory and autism. We're very interested in precision medicine. But one of these last things, which is very interesting, is that we're trying to actually make these human and animal studies much more aligned. And can we actually put them into the same machine learning model? And this is something that we're hopeful can be used to accelerate translational science. And so I just wanted to conclude that machine learning can be very useful to help accelerate fundamental understanding of neurological disorders. But I also think that if we can focus on how to make these things interpretable, how to make them explainable, we're going to be able to talk to domain experts and actually make much more progress on the disease, simple more than just prediction. And this is something called explainable artificial intelligence, which DARPA is pushing a lot of money into. And I'm really excited about what's going on. And I want to mention, I have lots of other projects going on. I wanted to only focus on one project today so that I don't just overwhelm people with everything. But I've also have projects ongoing on electronic health records, deep learning, environmental exposures, and so we have a lot of things going. And I think it's obvious from my talk for those but I should say it anyway. I think that a lot of these modern science and health applications really are going to require team science. If you saw from here, I have a lot of collaborators, and I think this is very necessary to make big impacts on these really large diseases. And I'm very excited about the future opportunities here, and especially the new Health Data Science Center, which I know Professor Pincina introduced a few weeks ago. So I'm really excited about all the opportunities here and where we're going to be able to take this. It's going to be great. And there's a lot of collaborators here. There's a lot of funding sources. This particular work was funded primarily by these three. And I wanted to end, so this is my final slide, but I wanted to use this slide in order to help lead us into the discussion part of this, which is, what is the biggest barrier to using machine learning in your own work? Because I'm actually very curious in the answer. And thank you so much for your attention today. I think there's a moderator somewhere. Okay. I was expecting a lot more of E. That's true. It's true. Oh, somebody heeded my wish.
You're probably familiar with some of the debate, especially in the more traditional settings where people talk about supervised and unsupervised machine learning. And so your example, not being the expert, I would classify it still as supervised learning. What's your view, and where do you fall on the spectrum, right? Because I, for example, have a lot of worry about the unsupervised methods where we don't give the labels, don't talk about cause and effect, and let the algorithms determine it. But can you push back and offer examples where in biomedical applications you would see value for unsupervised?
So that's a great question. I just want to elaborate on, so in these unsupervised applications, we're simply trying to find the structure of the data. So if we have a lot of data, we want to represent it, and we want to find interpretable trends and patterns without using any outcome information, which, as Professor Pincina pointed out, that can be rather dangerous because it's hard to figure out what's going on. So my example here is actually much more of a hybrid because we're actually using a lot of unsupervised formulation. So in the end, it turns into a supervised problem. But one of the things we're doing is trying to explain the data. To me, this is an interesting problem. So there are a number of problems in biomedical pre-processing that are completely unsupervised. So one of the problems I work on a lot is actually on what's called spike sorting. So in spike sorting, we want to find individual neurons. So as I mentioned in the beginning of this talk, we have implanted electrodes. We're going to pull out voltage signals. We're not pulling out individual neurons. we're pulling out voltage signals. So we have to turn these voltage signals into individual neurons. And it turns out this is completely done with unsupervised methods. And there are certain applications where there's really just no other way than to use an unsupervised method. But I think that if you don't have a particular reason to do unsupervised methods, you should really be doing them supervised if possible. However, I would say in a lot of health applications, you don't get purely supervised data. And in fact, there's something called semi-supervised learning. I don't know if people are familiar with semi-supervised learning. So what semi-supervised learning is, it's really a hybrid between unsupervised and supervised learning, the semi. And what it is it says, well, we may have a billion examples of just things without labels, but we don't have very many labels. And this is a much more common situation than you might realize. So if you want to go through and collect just lots of information, it's often very easy. But if you want to get outcomes, you often have to pay for outcomes, right? So there's a lot of benefits that people have started been showing, especially in image processing and image prediction, where we are using the semi-supervised approach, which is really a hybrid. So I don't even know how you would classify that. I think they're going back there.
Thanks, David. Great talk. So we have a question from an online viewer. And the question is it important to validate these results across multiple health systems, particularly for the EHR work that you're doing? And relatedly, does each health system have uniqueness that the methods or the data needs to be customized for each system?
So that's a fantastic question. And I think this is especially pertinent. And I think there's been a lot of debate right now, especially with the IBM Watson system, where people are claiming that IBM Watson does not generalize well to all hospitals. And I don't know, a lot of those results have not been published yet. That's just kind of the murmuring. And so I think it's very important to validate, yes, across all hospitals. And that becomes a very difficult question of how you do that while maintaining the privacy of individuals, which I don't think is completely solved.
Hi, I'm wondering kind of specifically about the collaboration between someone like you and an expert clinician. So if you just purely look at publications that come out, the vast majority of publications don't have named clinical collaborators. A lot of this work is often done solely in a technical domain. So being at a place like Duke with the Healthcare Data Science Center and your existing collaborations, can you highlight some of the things that you find most valuable for having close clinical collaborators?
Sure, that's also a great question. So I think what I really like in a clinical collaborator is someone that will push back on what I'm doing. I've worked with clinicians in the past who have told me that everything I do is amazing and wonderful. And while that's nice to hear, that's not actually very useful information because it's all positive, right? And so I really like somebody that pushes back and forces me to explain, because I think if they criticize parts of it, and especially in an intelligent way, I think it can really push the science so much further. But that just takes a while to build up. I mean, like when you first meet someone, it's really hard to understand the mutual problems that are going on. And so, but really what I like in a clinical collaborator is someone that just pushes back and really has their own ideas and will really push the boundaries of what we can do.
But I guess kind of as a follow-up, like right now, if you look at the survey results, like there's a lack of expertise. Like, how much understanding do you think a clinician needs of what's going on under the hood to be able to have those thoughtful, challenging conversations?
That's an excellent question, and I don't have a great answer for it. I know, for example, Jerry Dawson does not know much about machine learning, but she knows what we're trying to do, and she is very good at providing us feedback. Right, because she can ask us about our conclusion. She can ask us why we made the conclusion, what features were included. And so she has actually done a great job providing feedback, even though she does not understand the underlying machinery. And so, but where exactly is the level of knowledge you need to understand, I don't actually have a great answer for.
but that method seems not good. Afterwards, you are using the latent function. So I'd like to know, there's how many parameters, how many covariates you are using in the latent function? Is there any, how that correlated to your factor analysis factor?
So to repeat the question, I think there was a question about factor analysis versus we're actually using something called a latent function factor analysis. So it's still, I would still classify that as a suite of the factor analysis functions. The number of parameters that are unique to an individual window that we care about that are not marginalized out is still the number of factors we have, which was k equals 20. There are a lot of auxiliary variables that we just kind of collapse out. And then you have the number of variables that are needed for each of the individual factors. In a traditional form of factor analysis, the number of features per individual factor is 10,000 because it's a 10,000 dimensional object. For what we're doing, we structure it so it ends up being on the order of something like 700 per factor, which is still a lot, but that's way less than the 10,000 you would get naively.
One more, so thanks so much again, David. And we're actually right at time, but I'm wondering, for folks who have clinical questions that they think could benefit from a consultation or collaboration with your group and potentially applying machine learning methods, what's the best way for them to go about connecting with your team and brainstorming whether it's a good fit, and if so, what next steps are?
That's a great question. The easiest thing to do is to send me an email, but I think there needs to be a more formalized structure for doing this. And that's something that I think with the new Health Data Science Center that I hope we're going to be able to find a really nice way of doing that, because email only goes so far.